Okay, next we're going to talk about transcription. So, for DNA replication, we already learned that last year. So now we're going to skip over that and go to transcription. Transcription, memorize the definition. It's the process of copying a sequence of DNA to produce a complementary strand of RNA. So, in other words, it's a process where we use DNA to create RNA. And in translation, of course, there are three different stages: initiation, elongation, and termination. So initiation in English, it means start. So RNA polymerase, which is this pink round thing here, this thing, it looks at the DNA. That's the DNA, which is the gene. It looks at the DNA and it recognizes it and goes like, oh, there is the starting site. So it's the transcription starting site. This thing is called the promoter. So the promoter is kind of like the primer for DNA replication, but for here, it's called the promoter. Transcription starting site. It's where transcription starts. So a specific DNA sequence combined with like other proteins, it helps with the recognition of recognizing where to start, where the RNA polymerase should start. And what RNA polymerase is, it's an enzyme, and it helps bonds these nucleotides together, and it makes them to be attached together, and it creates this strand called RNA. And so this RNA polymerase, this thing, and other proteins that assemble on the DNA strand, they start like unwinding. You see here, it's unwinded, right? So this thing, this double helix is unwinded, and oh, I should have done that. Anyways, it starts to unwind this DNA double helix. The next stage is elongation. So this RNA polymerase, this thing is that it starts stringing together complementary strands of RNA nucleotides. So what it does is it starts attaching these nucleotides together, and see if you see here, if this is A, then this should be U, and if this is A, then this is U, and if this is G, this should be C, right? Because then the nucleotides, they should be complementary to each other, and the things that need to bond to each other will bond to each other. And the thing about elongation is that it only uses one strand of DNA as a template. So it uses only this one strand. It does not use two strands at the same time. It only uses one. And so RNA base pairings for the nucleotides, they are exactly the same as DNA. So G bonds with C, but the only difference is that U is the one that replaces T. So A, instead of binding with T, it starts binding with U. So A, U, and G, C. And so this growing strand of RNA, it starts to like hang freely, as you see here. As it's like transcribed, as it's like created, it starts to like kind of hang. And then the DNA, the DNA here, right? So as this RNA polymerase is moving to the right, the DNA is zipping back together, and it zips it back together. And in the end of termination, which is the last stage of transcription, so in here, the RNA strand here, it completely detaches from the DNA. So this thing right now, it's attached to DNA, but in this last stage, termination, it comes off. And so this RNA just pops off of this DNA, and it becomes a totally different thing. And this thing kind of zips back together. It becomes DNA again, which it was in the past, and this becomes RNA. So this happens when it reaches this place called a terminator region, where it goes like, okay, we stop here, and now this RNA will be detached from the DNA.